Chapter 10, Nutritional Genomics. Learning objectives. Define nutritional genomics. Discuss the Academy of Nutrition and Dietetics' position on nutritional genomics and the ethical challenges associated with genomics. Identify how an unfavorable environment for microorganisms can be created to prevent transmission. Explain the basic guidelines of universal precautions. Describe how polymorphism within genes occur. Understand different types of polymorphisms and their impact on health. Describe the regulation of gene expression and the relationship of epigenetic variation to developmental origins of adult disease. Discuss gene-environment interactions and their potential role in chronic disease. Recognize the challenges registered dietitian nutritionists engaged in nutritional genomics practice might occur. In 2003, the International Human Genome Sequencing Consortium published the finished version of the human genome sequence, thereby making a historical milestone in science. The human genome is the blueprint for over 20,000 different proteins. In many respects, the human body is a system of proteins. Proteins serve as structural components, hormones, neurotransmitters, and cell signaling agents that ensure the body is operating smoothly. This interaction between nutrients and other bioactive dietary components in the genome is known as nutritional genomics. Nutritional genomics further subdivided into the areas of nutrigenetics, nutrigenomics, and nutritional epigenomics. Nutrigenetics studies the role of the interaction between individual gene sequence variations, genotype, and dietary components in determining health. Interventions that consider genotype in conjunction with other factors such as current health status, food preferences, and other environmental factors may be referred to as personalized nutrition or precision nutrition. The field of nutrigenomics focuses on the influence of dietary components on gene expression and ultimately the production of proteins and metabolites. Figure 10.1 is a timeline of genetics and genomics from delivery by Mendel of the laws of genetics in 1865 to the completion of the Human Genome Project in 2003. This is figure 10.1 continued. Over the past few decades, various dietary guidelines have been developed for the purpose of optimizing health, preventing or treating cardiovascular disease, preventing cancer, treating hypertension, and treating diabetes. While based on the best available knowledge of the relationship between diet and disease, these guidelines do not take into account the genetic or epigenetic variations within the population and how that variation can play a role in an individual response to dietary factors and hence health and disease. Attention to nutritional genomics and pharmacologic economics have been escalated, and this trend is likely to continue in the coming years. In the Academy's position, it stated that registered dietitian nutritionists need basic competency in genetics as a foundation for understanding nutritional genomics. Proficiency requires advanced knowledge and skills. While Mendel, the father of modern-day genetics, discovered the laws of genetics in 1865, DNA was not itself identified as the blueprint of life until 1994, and its double hexyl structure was not discovered until 1953. DNA makes up the genome, which does not itself build an organism. From a human perspective, the genes contained in a person's DNA encode essentially the same proteins, but these code varies from person to person. The genetic material or genome lies within each nucleus of each cell in the body. In humans, the genome comprises 23 pairs of chromosomes. During mitosis, cell division within an individual, all 23 pairs of chromosomes are copied during the creation of a new daughter cell. During meiosis, reproduction, only one member of each pair of chromosomes is passed on to the ovum or sperm cell. The result is offspring that contain chromosomal pairs created by the donation of one copy of each chromosome from each parent. Of the 23 pair, 22 are autosomes and one pair comprised of the sex chromosomes. Males have one X and one Y chromosome, while females have two X chromosomes. So the gender of the offspring is determined by the sex chromosome passed on by the male parent. Each chromosome consists of DNA containing a linear sequence of genes, each encoding a specific protein. Figure 10.2, the chromosome 10 sequencing and analysis of human chromosomes have enabled researchers to characterize in detail a number of genes associated with diseases. Identifying the genes on all human chromosomes offers scientists worldwide an invaluable resource for improving human health and combating disease. Knowledge about genes will increase understanding of how genetic influences the development of disease, help researchers find genes associated with particular diseases, and aid in the identification of appropriate dietary interventions and development of new pharmaceuticals. Chromosome 10, which is pictured here, includes 700 to 800 genes and has been linked to various disease states with nutritional implications, including several cancers, type 1 diabetes, and Crohn's disease. Figure 10.2 is a nucleotide with the DNA molecules. The four nitrous bases of DNA are arranged along the sugar phosphate backbone in a particular order, encoding all genetic information for an organism. The genetic code is the code responsible for translation of DNA into protein, was identified as a triplet code in 1961. A series of three nucleotide bases. The codon encodes a specific amino acid. DNA also contains non-codon regulatory sequences called promoter region to which molecules can bind in order to signal underwinding of a specific region of DNA for creation of a needed protein. Over 85% of DNA in humans is made up of non-coding DNA, which lies within or is between expressed genes. Transcription is accomplished by the enzyme RNA polymerase, which first complexes with transcription factors in a gene promoter region before facilitating production of mRNA. The DNA strand that serves as the template for mRNA synthesis is known as the sense strand, whereas the non-codon strand is the antisense strand. Once transcription is completed, mRNA undergoes post-transcription processing. Enzymes in the nucleus excites segments of the mRNA known as introns, intervening sequences, while leaving the segments known as extrons, express sequences. During translation, the triplet codon comes into play. Most amino acids have multiple codons, but each codon only encodes one specific amino acid. Small molecules of another of the RNA transfer RNA serves as an anticodon. After the amino acids are positioned in sequence, peptide bonds are formed between adjacent amino acids, and the new protein elongates until a stop, a nonsense codon, is reached and nearly created proton is released. Additional processing of new proteins is called post-translational modification. Figure 10.4 is transcription and translation. When genes are expressed, the genetic information on the DNA is first transcribed to a molecule of messenger RNA in a process similar to DNA replication. Polymorphism variations exist within genes throughout the population. Most of these variations are not cause for concern. The outcome of a given variation depends on the nature and location within a given gene. In other words, a specific variation may have no effect on the production and function of the protein product. It is also possible that a single nucleotide change or a more complex alteration in a single gene can have profound effect. Figure 10.5 DNA sequence variation in a gene. Specific codon direct the cell's protein synthesizing machinery to add specific amino acids. Inheritance of specific genes can be classified as autosome dominant, autosomal recessive, X-linked dominant, X-linked recessive, or Y-linked. Because individuals inherit one copy of each gene from each of their parents, the actual expression of an inherited gene can vary, and the gene expression is what determines phenotype. For example, brown eyes are autosomal dominant, whereas blue eyes are autosomal recessive. If an individual inherits the gene from brown eyes from one parent and the genes for blue eyes for the other parent, his or her eyes will be brown because that is the dominant gene. Whether a trait is recessive or dominant determines whether that trait is phenotypically expressed. When the alleles from each parent differ from each other, as in this case, an individual is heterozygous for the gene. Understanding monogenetic disorders such as those described earlier helped lay the groundwork for comprehending the complexities of polygenetic diseases such as obesity, diabetes, cancer, and cardiovascular disease. The study of gene-nutrient interactions that are dependent upon gene variances is focused primarily on single nucleotide polymorphism. SMP are defined as those genetic variants or polymorphism in which a single nucleotide is present in place of another. Epigenetics relates not to the genome sequence itself, but to the inherited patterns of genes expressed, regulated by modifications to DNA. Gene expression is related in many ways, Although humans have the full genetic material of all nucleated cells, not all genes are expressed in all cells, and the actual level of expression varies based on DNA. Individual SNPs may not be the best measure of genotypes. Regardless of individual genotypes, environmental factors play a large role in regulating epigenetics and therefore gene expression. In other words, diet, activity, smoking, and so forth can turn specific genes on or off and thus determine the quantity of specific protein product produced as well as the activity of related metabolic pathways. Several well-defined and relatively rare cancers have a clearly established genetic inheritance based on the mutation in a single gene. An inherited mutation, for example, carries 1,000% risk of developing the disease familiar. Predicting benefit is difficult because people do not all respond in the same way to environmental exposures. Obesity has a clear link to genetics, demonstrated by numerous studies showing that obesity does persist in families even where food intake and physical activity patterns differ. However, the escalating obesity epidemic in recent decades support the idea that while susceptibility to obesity is genetically and epigenetically determined, the development of obesity itself is a result of susceptible genomes in the presence of conducive environment. 12 genes to date have been identified in monogenic obesity syndromes, primarily in severely obese individuals with onset at an early age. Hundreds of genes have been examined for potential roles in the development of type 2 diabetes, including those that may play a role in a pancreatic beta cell function, insulin signaling, and so forth, in order to identify targets for precision nutrition intervention. Examples of interaction of dietary components with the genome modulation of lipid metabolism, inflammation, and development of cardiovascular disease are many. Early studies examine relatively simple relationships between individual SNPs and single nutrients. Registered dietitians must be knowledgeable in general genetics and genomics concepts. They must also be able to understand the toll of diet in interaction with the genome, knowledgeably read and interpret the current research literature, and place the role of the nutrition genomics in context with phenotype characteristics. Beyond evolution in clinical practice, there will be other changes as well. Research will continue to identify bioactive food components and examine how they interact with specific genes and specific genotypes and genetic risk scores, and how they influence epigenetic type and gene expression to yield changes in health risks.